Hey guys, I wasn't able to make class on Tuesday. I know the topic is genetics, but I was just wondering if y'all could fill me in on what Mr. Dr. Mutar covered.
He kept saying something about the genomes.
That's real funny, Kyle.
So I know that genomics is the basically the architecture of genetics, and kind of covers the function, the organization, the content of genetics. But what else did he go over as far as like structural and functional genomics, mapping, some of those details?
Yeah. So as far as structural genomics goes, we talked about the Human Genome Project. It's pretty self-explanatory. I don't think you need me to explain it to you. You know, sequence the genome, that whole thing. Yeah, so it started in like 1990, ended in 1993. Then he talked about some of the benefits, medical benefits, understanding the genome, drugs that could be tailor-made to genomes, helping address cancers, better prognosis for cancers. Also, apparently, we're all like 1 to 4% Neanderthal. David, I think you may have a little more than me. I'm just playing. Yeah, so then he also talked about how we sequence the genome, how we sequence the genome. We used initially map-based sequencing, and with the map-based sequencing, we used two different types of maps. We used genetic maps and physical maps. Genetic maps are based off of recombination, and they're like measured in recombination frequencies, and they're not as accurate. And then there were physical maps, which was specifically analyzing the DNA, directly analyzing the DNA. So a physical map would be like a map to your neighborhood with the specific houses and their addresses. And then a genetic map would be like a larger city map that was more generalized and less specific, not quite as accurate. We also talked about SNPs, SNPs, single nucleotide polymorphisms. It's just a difference, a mutation at one base pair. And talked about how it's not always seen in the phenotype. We talked about copy number variations, which are can be deletions or duplications of large numbers. Can be like hundreds of thousands of base pairs of DNA. What else did we talk about? Haplotypes. We talked about haplotypes, which are a specific set of single nucleotide polymorphisms on a chromosome or part of a chromosome. I think that's everything, at least for structural genomics. Yeah. Also, I called you a Neanderthal, David. I feel bad about that. That wasn't very nice. I just mean that you look like you eat a lot of protein. Like you have bigger muscles than I do with these little arms.
Oh, thanks, Sean. Yeah, just eating a lot of protein. Does that go along with proteomics? Do I need to study heavier on proteomics so I know how much protein to eat? Did he go over the importance of microbiome? How specifically did he talk about that? Microbiome, and then the method for studying microbiome, metagenomics, and looking into a whole group of organisms that share a common environment. What exactly did he have to say about that?
So he didn't really go over any microbiome stuff. I don't think that's really relevant for what we're doing now. And no, David, proteomics is not about how much protein to drink in your shakes. That's a whole different ball game. But so basically what proteomics is looking at the particular structure and function of proteins and how they're kind of categorized. So basically we can take proteins and separate them with SDS page or 2D page, which is basically electrophoresis. So the SDS is detergent that's going to break down the protein, and then we can basically separate it into its individual peptides and see what it's doing, and then we can basically put those onto a spot or a microarray and see how those different proteins are being expressed at different in different regions or for different reasons. Kind of along the same lines with that is going to be the transcriptomics, where basically you do the same thing almost, but we do that with express sequence tags or ESTs. So basically we're going to get some transcripts from both normal tissue and cancer tissue and see and tag those transcripts, and then put them through database and put them through kind of a microarray technology again, and you can do this with two-dimensional. Let's see if I can do this. So like that two-dimensional array. So basically the red is bad and the green is good for the most part, or green is down-regulated, red's up-regulated. So we can see that there's different parts that are regulated, different sequences. So that can kind of give us an idea of where, at what stage of the cycle is producing these changes within the transcript. Hope that helps.
Thanks, guys. Yeah, that was super helpful. So basically going off the rough definition that I gave, that is that genomics is generally the content organization and detailing the function of the genetic material of genetics generally. Sean, you broke it down, I guess, for me from the structural genetics side, talking about map-based sequencing and how map-based sequencing breaks down into genetic maps and structural maps. And between the two of those, structural maps are more specific and accurate, as they give it like a physical location within structures, as to what we're looking for. And and then Kyle, you broke down functional genetics, which goes into detail of transcriptomics and proteomics. So transcriptomics kind of being the again referring back to the architecture of the RNA molecules transcribed by a particular genome, and so what RNA molecules are actually transcribed, and then detailing those, and then the same thing with proteomics, the proteins that are translated by a particular genome in sequence. I think that covers it. Thanks, guys, for your help.